And so we come to some groundbreaking work that won the Nobel Prize for Medicine and Physiology in 1981, done by Hubel and Wiesel. In these studies, they made use of a new kind of technology that had just become available in the late 1970s. And this was the ability to record the activity of a single neuron in the living animal. Now, to do this, you need a very, very, very fine electrode. The very first ones were handcrafted out of glass, actually, made absolutely tiny, and they were soon replaced by titanium electrodes, which were a little bit more robust. And once this was available, what this experimental sequence did was study the activity of individual neurons in and around the primary visual cortex in a cat brain. Now, the cats were anesthetized, so they are unconscious cats with their eyes propped open, pinned down on a lab bench, and then things are flashed on the screen. So this is what it looks like, a light bar stimulus projected on the screen there. The cat has its eyes open, and the stimulus can be varied until you find something that seems to correlate with the activity of an individual cell. There on the right-hand side in the column, you can see that the vertical bar in this case seems to be eliciting a strong response, whereas a horizontal bar doesn't. And so there seems to be a sensitivity of this particular neuron to the presentation of a bar at a particular orientation in the visual field. Now, a bar is a very simple element. It's not going to be possible to represent much with that. But the story develops then with the much older idea that simple elements might be extracted from the sensory information and then combined initially within a mode and then between the modalities in order to produce whatever it is that is produced in perception, a mental representation or a mental state or means of control. What they found, furthermore, was that as they moved away from the primary visual cortex, which is the first stopping point in the brain after the retina, and moved into the depths of the central nervous system, what they found was neurons that responded to much more complex patterns of light and dark, or stimuli. And with this, the idea became strongly developed that vision could be understood as the progressive construction of a representation of the world based on sensory primitives. Now, identifying the stages in this turns out to be a non-trivial fact, and in fact about half the neurons in the brain co-vary with what you see, so there's some visual components to an awful lot of brain activity. Shown here are some complex sequences of hypothetical stages here, and it gets very difficult to trace things after this because the brain is not a feed-forward system in which one area simply projects to another area. It's a massively recurrently connected system in which lots of things are connected to lots of things, and pretty much everything is connected to everything else. Nonetheless, visual anatomists and physiologists in this area have identified distinct areas of the brain that seem to co-vary with different goings-on in the visual field. And that leads us into our second of these major studies, which is by Goodale and Milner. Proceeding in a fashion not unlike Hubert and Wiesel, but somewhat later, they identified what they described as two processing streams starting in primary visual cortex. This is a cat brain, incidentally. Primary visual cortex is area V1 there on the right-hand side of your screen. But they could follow two distinct paths from here, one called the dorsal stream and one called the ventral stream. And building on the ideas of Hubel and Wiesel, they suggested that as you progress along these streams, more and more information is combined from different sources, including high-level knowledge, expectation, multisensory information, and so on, in order to give an increasingly powerful and useful representation that can guide action. So let's have a look at these two streams, the dorsal stream and the ventral stream. The dorsal stream comes up over the back of the back of the brain, up to the parietal cortex, sometimes also called the where stream, as this stream seems to harbor neurons which are particularly sensitive to motion, to movement. Now, movement here must mean change in the visual field, which can either come about because things in the world move, or can come about because you move. So this kind of information that is very sensitive to movement, is necessary in order to keep track of things in the environment, and also to stop bumping into lampposts, to guide action, to guide movement. And that seems to be the principal concern of the dorsal stream, and this is undoubtedly something that we would want from a visual system. One of the uses we put the visual system to is to avoid bumping into things. So what about the ventral stream? The ventral stream projects down into the temporal cortex, inferior temporal cortex. And this is sometimes called the what stream, because as you examine the properties of neurons here, they seem to be not concerned with movement, but with the identity of things. That is, the ability and capacity to recognize, name things, categorize things, seems to, this is another thing that vision does for us. And it seems to be a somewhat separate task from that of not bumping into things. So we've got the where stream and the what stream, concerned with movement and with recognition. Now, let's just have a look at what this kind of work looks like. Here's a brain, and the red bit there is the inferior temporal cortex, which is the terminus, as it were, of this ventral stream. And here you find, when you go in and you take your very, very fine electrodes, you find neurons that seem to respond selectively to specific objects or complex shapes. There are some examples, bananas or a circle with a T shape attached to it, very complicated things. And it's very difficult to know in advance what any given neuron will respond to. The way this work goes is you prepare an animal, usually a monkey. You insert an electrode into one of these neurons, and then you show it things, and you've no idea, you've no way of systematically searching the space of all possible things. So by good luck, you may find a neuron that responds to something, but it's quite difficult to understand what its neighbors are going to be, even when you figure out what one neuron is responding to. So here's an example of two such studies. On the top left, you see a plot. At the top, you see a series of monkey faces from full frontal to profile. These were shown to the experimental subject, who was also a monkey, so these are conspecific faces. And when there is an electrode in a single neuron, and it has been found that this neuron responds selectively and preferentially to faces. And if you look at the responses, either the histograms in the second line or the aggregate response in the bottom line, what you see is that this neuron seems to be particularly active when the monkey is looking at the face of a conspecific in profile. Not even head on, but in profile. That's a remarkably specific kind of sensitivity to find in a neuron. At the bottom right, we can follow some of the kinds of questions you ask when you find such a neuron. So this is a neuron whose response is shown in the red histograms up there, and we start at the top left. This neuron responded strongly to a picture of a hand like this. So then you start varying the stimulus to see, well, what is it about the hand that seems to be important? In other words, how does this neuron understand a hand, or what kind of hand is it sensitive to? So one thing you can do is go from this to this, turn the hand around, and we find it still fires. Then we can replace an actual hand, which has lots of texture and detail, with the mere outline of a hand, and we find the neuron still responds to it. So it's something about the shape of a hand it's responding to. Furthermore, we can turn the stimulus from this orientation to this orientation, and we find the neuron still responds. So that generates a completely different pattern on the retina, but that's irrelevant to the identity of this thing as a hand. Then we can shrink the hand, look to see is it not only rotation invariant, but size invariant. And what we find is that this neuron is not size invariant. That is, as the hand gets smaller, the image of the hand gets smaller, so the response of the neuron gets weakened. Then we can vary the hand in other ways, so we'll replace it with a glove-like outline and the response goes away. So the fingers are really important. Then we're going to start in the bottom line. Here we try various things with fingers that aren't hands, and we find that the neuron doesn't respond to them at all. We try faces, shapes, and so on. So, having explored all this, one might ask, what is this neuron actually sensitive to? And here's a question for you, which I think you might be able to answer. Who owns the hand that this neuron is sensitive to? It's a bit like asking what did the bus driver have in his sandwiches, except you actually have enough information to answer this. Consider your own body and your own environment. Look around you. You may see hands somewhere off in the distance, up close and so on. But there's two hands that are very special. Your hands, because they tend to be at a fixed distance from your head. When you manipulate something, you do so in a dedicated space in front of your body. So this neuron is probably responding to the sight of the monkey's own hand. Now, these are very, very specific responses, and as you can see, it's not clear how to understand where a hand fits in here. So shown at the bottom left there is just this general notion that although there is some kind of systemic organization of neuron properties here, they're very, very complex, and it's very difficult to say what the basis for that organization is. So as we ascend these pathways, cells are selectively responsive to more and more abstract properties. And there was a sort of an urban myth or a fictitious goal of cognitive neuroscience, which would be to find the cell that represents your knowledge of your grandmother. That would be the cell that fires only when your grandmother is present and not otherwise. Nobody's ever found that. But they have found cells which are sensitive to very high order properties, very complex combinations of features. For example, one patient who was about to undergo brain surgery was tested. You only do this before surgery because you're sticking needles in people's brains. And they found a cell that responded selectively to Jennifer Aniston's face. I kid you not. That doesn't mean it's the grandmother cell, and who knows what else that cell responded to, but it had that specific character. We can illustrate this quite graphically by using something called the Thatcher effect. The Thatcher effect tells us something about the manner in which we recognize faces. Faces have parts. Glasses, eyes, ears, hair, and so on. But they also have an overall configuration. And furthermore, I don't know if you've noticed this, but we tend to see faces with a particular orientation, right way up. We rarely see upside down faces. So here's an upside down face. This is Professor Brian Vonson in the physics department in UCD. And his image here, I don't know if it strikes you as odd. It should. It's been manipulated. It's very, very odd. If I turn it the right way up, you'll immediately see why it's odd. It's actually very, very grotesque. It's ghastly. Back. Forward. What's happened is the eyes and the mouth have been photoshopped and turned upside down. So when you look at it this way, you're seeing all the pieces of a face, but you're not really seeing the face as a whole. That is, there's something missing, so that seeing faces seems to involve more than just identifying parts of a face. It also seems to require the recognition that these elements are in a particular configuration, and for that the face needs to be right way up. We can illustrate this again. Here I can rotate this picture, and you can observe for yourself that there is a definite switch. It's currently grotesque, and now it's not grotesque. And we can do this all day. And the point at which it switches from non-grotesque to grotesque and back again will never change. This is not a matter of learning. This is a matter of neural circuitry. So this effect is called the Thatcher effect after Margaret Thatcher, whose face was first used in this. She had nothing to do with the research. Right.